Welcome to Part 2, Health and Hygiene. What is biological contamination? How does food worker health affect safety? What should you do when you're sick? And what does good personal hygiene mean? Find the answers to these and other important food safety questions, including proper hand washing and when it's important, how to avoid bare hand contact with ready-to-eat foods, glove use, and how personal habits can affect food safety.
Of all the types of hazards that can arise from unsafe food handling practices, biological contamination is the most common. Germs are everywhere, and some of them make us sick. It's those harmful pathogens that cause foodborne illness.
Food worker health is one of the most important factors to preventing foodborne illness. It's simple. When you're sick, don't work with food. Germs can easily be spread if you're experiencing flu-like symptoms, jaundice, or other illness.
If you're sick, stay home. Remember the 24-hour rule. Vomiting, diarrhea, fever with sore throat, or jaundice, stay home for 24 hours from the last symptom.
It's important to report these symptoms to your PIC, as well as if you have an open wound or sore.
If my employees are sick, I tell them to go home and come back when they feel better. It's not worth the risk.
Besides being aware of your own health and knowing that when you're sick, you pose a risk to the public, being aware of your personal hygiene is another aspect of avoiding contaminants in the food you serve. In fact, food workers with good personal hygiene save lives. Following proper hand washing protocol by washing your hands the right way and at the right times, keeping fingernails trimmed for easier cleaning, wearing hair restraints and proper work clothing, and covering cuts and burns with bandages and gloves, in addition to not working while sick. These are all ways that hygiene prevents the spread of germs in the kitchen.
Hand washing is something we all learned at a very young age. But if you want to work as a food handler, it's important to relearn this task in the proper way that complies with the health department guidelines. But first, let's review when it's important to wash up.
Wash your hands throughout the day, even when hands look clean, to keep germs off your body and the food you prepare. Always wash before you begin food preparation, or if you've been contaminated by exposure to potential germs. Contamination can be caused by using the bathroom, touching one's face or nose, handling raw meat, fish, or poultry, sneezing, coughing, or blowing one's nose, handling garbage or dirty dishes, handling animals, or using chemicals, or after taking a break, eating, or smoking. If you aren't sure if a wash is necessary, wash anyway, just to be safe. Proper hand washing is a 6-step process that requires a hand washing sink with hot and cold running water, soap, and paper towels or other single-use drying method. Step 1. Get hands wet to help the soap work. Step 2. Apply soap and scrub. Remember to pay attention to your entire hand when washing. Step 3. Scrub your hands together for 10 to 15 seconds. Step 4. Scrub the backs of your hands and between your fingers, a common breeding ground for bacteria. Scrub your forearms. They frequently touch food prep surfaces, and utensils. Step 5. Rinse hands to send the soap suds and germs down the drain. Rinse for about 5 seconds to fully remove the soap. Step 6. After rinsing, dry hands and forearms using either a disposable paper towel, air dryer, or a continuous cloth towel system that supplies a clean towel each time. It's important to know that hand sanitizers may be helpful on clean hands, but are not substitutes for hand washing. In fact, in some cases, double hand washing may be required. Always remember that hand washing is the most important food safety tool to get rid of the germs that make people ill. When in doubt, wash. But germs can remain on your hands even after washing. That's why it's important to prevent bare hand contact with ready-to-eat food by using utensils such as tongs, scoops, deli papers, or single-use gloves.
Focus on minimizing bare hand contact. Utensils, tongs, tissue paper, these are all acceptable when handling ready-to-eat foods. So you don't need to glove up as often as you think. When you do use gloves, remember it is required to wash your hands before and sometimes after proper glove use.
Ready-to-eat foods are those served without additional washing or cooking to remove germs. They include washed produce that's eaten raw, such as sliced fruit, salads, and garnishes. Foods that will not be cooked, such as sandwiches, sushi, and deli salads. Bakery or bread items, such as breads, cakes, pies, and tortilla chips. Ice that may be used in drinks, or foods that have already been cooked. Gloves can be tricky because if a food handler thinks they're using gloves correctly and they aren't, they can put the public at an increased risk by contamination. Gloves are there to protect food from germs, not your hands from the food. So remember to change them often and never wash or reuse them, especially between working with raw and ready-to-eat food. When you're done with them, be sure they go in the trash and wash your hands afterwards. When it comes to health and hygiene as a defense against foodborne illness, it's important to realize that personal habits affect food safety. You may not eat, drink, or use any type of tobacco in food prep areas. This is to prevent spills and reduce contamination. Use hair restraints that are intended to keep hands out of hair and hair out of food. Hair must always be restrained when working around food or in food prep areas. Hair restraints include hairnets, barrettes, ponytail holders, and tight braids. Long beards must also be restrained. Fingernails must be trimmed for easier cleaning. If nail polish or artificial nails are worn, you must wear gloves when preparing all foods, not just ready-to-eat foods. Rings, watches, bracelets, and all other jewelry on arms or hands must be removed during food preparation. Wedding rings may be worn if they're covered with the glove when the food worker is preparing food. Personal items like medicine, coats, and purses must be stored away from food, dishes, and linens. Let's review Part 2, Health and Hygiene. We learned that biological contamination is caused by germs on our bodies and in our environment, and that if you are sick, don't come to work. Follow the 24-hour rule and check in with your person in charge about your symptoms. Follow proper hand washing steps at the correct times to avoid biological contamination. Avoid bare hand contact with ready-to-eat foods. Use gloves to protect food, not your hands, and wash before and after glove use. Be aware of your own personal habits and hygiene as a defense against foodborne illness.
What makes beer? Single-celled organisms. What makes us cough and feel like sleeping for 12 hours straight? Single-celled organisms, also known as microbes, germs, the tiny machines that allow us to digest and kill us. What is life? Turns out life is relentless everywhere on Earth. Microbes are invisible, they're single cells. So to understand microbes, that is to do microbiology, scientists first needed to study cells. German botanist Matthias Schleiden and German physiologist Theodor Schwann proposed a modern cell theory around 1837. Every living thing is made up of one or more cells, so the cell was the fundamental unit of life, the thing that gives structure to all forms of life. Amazingly, Schleiden, who studied plants, and Schwann, who studied animals, both noticed tiny dots in the middles of the cells they looked at under their microscopes. These nuclei must somehow be important to all life, they figured. correctly. Darwin's chief public defender, Thomas Henry Huxley, also wrote an article called The Cell Theory in 1853. Where the Germans were cataloging different microscopic forms, Huxley proposed a mechanistic model. Cells are little factories with different parts that have different functions. Their functions make the parts add up into an organic whole. Not every scientist believed cell theory right away, but lots of them came around as microscope-enabled evidence mounted. And yet, being able to observe single-celled organisms didn't immediately lead scientists to connect them with disease. In fact, medicine in the early 1800s mostly worked the way it had hundreds or even thousands of years earlier. It was still all about liquids called humors. But some diseases were understood differently. They were caused by an invisible badness in the environment called miasma. According to miasma theory, diseases were caused by foul-smelling airs that came from rotting meats, swamps, and other putrefying sources. Before you knock it, think about it. This theory mostly works. Dangerous microbes grow on smelly, dirty stuff. The smell is a byproduct of the microbes eating organic matter. But even today, we can't see the microbes without instruments, so following our unaided senses is still smart. Don't eat gross trash. Sniff the turkey before you eat it, and then just eat it anyway, like I did this morning. By the mid-1800s, however, a few scientific rebels were pushing a germ theory of disease. This holds that germs, or pathogenic microorganisms, cause infectious diseases. Around this time, a nasty germ called Vibrio cholerae provided an opportunity to test the theory out. Cholera had ravaged England since the germ started to travel widely out of South Asia in the early 1800s, hitching rides on the expanding armada of merchant ships. Empire had all kinds of unpredicted downsides. In 1854, a bad cholera epidemic hit Broad Street in the Soho neighborhood of London, killing over 600 people. One doctor, named John Snow, had a hunch about what was going on. Snow created a map of the outbreak and noted that the cases seemed to cluster around one public well. He hypothesized that cholera microbes were rapidly multiplying in its water. So he convinced the Soho Council to remove the well's pump handle. People went elsewhere for their water, and cholera infections decreased. John Snow became a hero, saving lives, even though it turned out the outbreak was already in decline when he removed the handle, thus proving that some John Snows do know some things. John Snow's quick thinking didn't really prove germ theory. For germs to replace miasmas in their minds, scientists would require a better model of what germs are and how they behave. For that, we turn to one of the first experimental microbiologists, and a dude whose name you probably should know from milk labels, Louis Pasteur. Born in France in 1822, the same year as Galton, Pasteur was a chemist, not a physician, but he is known for his work on germs and health. Pasteur is credited as the co-founder of microbiology and the founder of modern zymology, the science of fermentation. Fermentation is the biological and chemical process needed to make beer, wine, yogurt, cheese, bread, lots of other foods. Thought bubble, show us what that means. Chemically, fermentation is the conversion of sugar molecules into ethanol or alcohol and carbon dioxide. In food, this usually happens thanks to bacteria or yeast. Looking good, Saccharomyces. Pasteur first connected yeast to the fermentation of wine in 1857. He observed that microscopic yeast on grape skins makes grape juice ferment into wine. If you sterilize the skins, killing the yeast, fermentation won't happen. Pasteur even made the analogy that the microbes growing in wine and beer were similar to the microbes causing diseases in humans and animals. He hypothesized that microbes are everywhere and must be responsible for all kinds of phenomena, like making food spoil. So in 1865, he patented a process in which liquids, such as milk, were heated to a temperature between 60 and 100 degrees Celsius in order to kill microbes. This became known as pasteurization. This might sound kind of simple, but it was revolutionary. Earlier, Nicolas Appert had invented canning to make food safe, so Napoleon could fight wars for longer, but Pasteur's method used a lower temperature, thus preserving tastes and textures. Related to this idea of killing bad germs, Pasteur also helped develop hygienic medical practices, along with a British surgeon, Joseph Lister. No germs in the operating room. Most importantly for the history of biology, Pasteur got tangled up in a debate with a fellow scientist named Félix Pouchet, who believed that living things spawn naturally from certain environments. This widely held belief was called spontaneous generation. Pasteur, based on his observations of grapes, reasoned that living things can only grow from other living things. So he created an ingenious experiment to prove the theory of biogenesis, or life from life. Pasteur showed that no germs would grow in sealed, sterilized, long-neck flasks full of beef broth. But in sterilized but open flasks, microbes would eventually show up. Thanks, Thought Bubble. After proving biogenesis in the 1870s, Pasteur worked on immunology and vaccination. Pasteur's work with the rabies and anthrax microbes supported the germ theory and its use in medicine. If you could identify a microorganism, you could give someone a weakened version of it, or vaccine, and the patient taking the vaccine would develop immunity to the real bug. And in 1879, Pasteur developed the first lab-manufactured vaccine for chicken cholera. It was about this time that germ theory was finally widely accepted, in part due to Pasteur's work. But Pasteur's ethics were questionable. When rabies vaccines for humans rolled out, Pasteur kept his data on their effectiveness secret. He even kept secret mortality data, or how many people his vaccines didn't help. So unlike today, in Pasteur's time, it wasn't necessarily a norm that useful medical research should be shared publicly. But Pasteur was not the only notable germ wrangler. Born in 1843, the German physician Robert Koch painstakingly worked at a microscope to definitively establish the germ concept of disease. He identified different microbes, becoming the leader of a large research team in Berlin. They founded the discipline of bacteriology, or the study of bacteria, particularly in relation to human health. In the late 1880s and 90s, Koch and his team traveled into areas experiencing epidemics, including India and Egypt, to identify the microbes causing the diseases there. By IDing these microorganisms, they hoped to create vaccines. First, Koch proved that the anthrax germ causes anthrax the disease. Later, he identified the bacterium that causes tuberculosis, or TB, which was a very widespread disease. Koch also characterized cholera, vindicating the work of John Snow by showing that the bacterium lives naturally in the small intestine of humans. Snow wasn't just high on ether, he had correctly guessed how cholera spreads. Eventually, Koch's team contributed to the identification of the germs that cause diphtheria, typhoid, pneumonia, gonorrhea, meningitis, whooping cough, tetanus, plague, leprosy, and syphilis. I would not want to borrow this dude's lucky microscope. It's been through a lot. Through this work, Koch developed the 4 postulates, or steps, for identifying infectious diseases. 1. The microorganism must always be present in every case of the disease. 2. The germ must be able to be isolated from a host containing the disease and grown in a lab in a culture. 3. Samples taken from the culture must cause the same disease when introduced into a healthy model animal, like a mouse. 4. The germ must be isolated from that new animal, meaning it's the same organism that caused the disease in the original host. So structure matters. In all infections, germs grow out of hand, throwing the body out of whack and causing inflammation. These bad germs, or pathogens, develop and live within us and can kill us. But with Pasteur's vaccines and Koch's scientific method of IDing bacteria, humans could now develop medicines to fight jerk sauce germs. Go team us! Back in the world of multicellular organisms, another scientist at the intersection of biology and medicine was exploring the structure of living things. In 1892, German-American physiologist Jacques Loeb began experiments on embryonic development, or how organisms grow from single-celled embryos into whatever they're supposed to look like. So he started dunking sea urchin embryos into different salt solutions, and in 1899 he got one to give birth. Just from being salty. Loeb's artificial parthenogenesis, or birth from the self, was mega astounding news. It raised basic questions like, what is sex? Could a human one day give birth by herself or himself? Causing sea urchins to give virgin births made Loeb a celebrity. But for the history of biology, it was revolutionary that Loeb's sense of scientific understanding of life was tied to his ability to control it. His work with sea urchins can be seen as an early form of bioengineering, or treating cells and tissues as machine parts to be used constructively by a human designer. Loeb's notable students included the behavioral psychologists B.F. Skinner and J.B. Watson, and a creator of birth control, Gregory Pincus. Biology, like other scientific disciplines, shows a historical arc from observations to experiments, from less control to more. From the mid-1800s to the early 1900s, scientists moved from mostly believing that living things spontaneously appear out of nowhere to controlling fermentation on a massive scale. And cheers to that. Next time, we'll add the missing mechanism to this crazy Rube Goldberg machine we call biology. It's time for a monk named Mendel to accidentally figure out genetic variation in mice and men and peas. Crash Course History of Science is filmed in the Dr. Cheryl C. Kinney studio in Missoula, Montana, and it's made possible with the help of all of these nice people, and our animation team is Thought Cafe. Crash Course is a Complexly production. If you want to keep imagining the world complexly with us, you can check out some of our other channels like SciShow, Eons, and Sexplanations. And if you'd like to keep Crash Course free for everybody forever, you can support the series at Patreon, a crowdfunding platform that allows you to support the content you love. Thank you to all of our patrons for making Crash Course possible with your continued support.
A pathogen is just a fancy word for a disease-causing organism, and there's a few different examples of them you should know, like virus, bacteria, protoctist, and fungi, and you should have an idea of an example of each one. Flu is one of the most famous examples of a virus. Cholera is an example of bacteria, as well as things like E. coli, which causes food poisoning. Malaria is a protoctist. That's the one that people most easily forget, are the protoctists. And then fungi are things like athlete's foot. In a transmissible disease, this is a disease in which the pathogen can be passed from one host to another. That's just a fancy way of saying it can be spread. It's a disease that can spread between things. So there are different ways that it can be spread or transmitted. It could be direct transmission, and that would be things like contact between people when they touch, mixing of bodily fluids, that sort of thing. And then also indirect methods, and this would be via contaminated foods, via animals. Some things might be airborne, or they might be spread through water, this type of thing. We've got something called vectors, and a vector is an animal that carries a pathogen from one host to another. A good example is mosquitoes. There are certain types of mosquitoes which can spread malaria. You know that bats can spread rabies, as well as other animals can spread that as well. A lot of different ways to reduce the transmission of disease. The first and most obvious one is hygienic food preparation. Obviously, wash your hands before touching your food, or wear gloves if in the food industry, if you're working with food, wearing gloves would often be a requirement. Keep your hair out of the food. That's a good way to spread things around, so keeping your hair out of it is a good way to prevent transmission. Don't cough or sneeze around the food. Keep animals away, and that includes flies. Flies are really good at spreading disease. Keep food cold, because cold conditions are not very good for bacteria and other microorganisms to grow. And also keep raw meat separate, because raw meat is a good location to harbour lots of pathogens. And when the meat gets cooked, then the pathogens get killed. But if that raw meat has been kept away from, has been kept next to something that is then going to be eaten raw, then obviously the pathogens might be on that raw food and can still be spread. Another method is personal hygiene. A lot of oil and dirt accumulates on the skin throughout the day, and obviously that means that pathogens are also going to get trapped within that. Also, sweat encourages bacteria, so washing regularly is a way of reducing transmission. Next would be waste disposal. Obviously, rats and dogs and all that sort of thing are attracted to food waste, and it's a really, really good place for bacteria to multiply. All of that leftover food waste is potential nutrients and potential fuel for bacteria. So well-designed landfill sites are essential, and also things like taking out the rubbish regularly from the kitchen, not leaving it to build up for too long, that sort of thing. The other thing is sewage treatment. Obviously, toilet and kitchen waste contain quite a lot of pathogens. If somehow sewage managed to enter drinking water, that's a really obvious pathway for transmission. So correct treatment of sewage is essential to make sure that the water that leaves, the dirty water that leaves, isn't then isn't then going to be able to enter the water that somebody consumes. Let's look at the sort of exam question you might get on this. How about describe and explain the ways in which a pathogen can be transmitted from one host to another? And that's a 4-mark question. First off, let's look at the command terms. First off, we've got a describe and then we've got an explain. So there are 2 requirements in the answer. And let's look at it in that context. First of all, pathogens can be transmitted through the air. That's the description. Next, a person can breathe the pathogen in via the lungs. That's the explanation that goes with that description. Next one, it could be transmitted via the blood. Let's explain that. In blood transfusions, bloodborne pathogens can transfer from the body of the first host into the body of the new host in the blood. Next one, let's describe that it can be transmitted via contaminated food, and then explain that. For example, pathogens are transferred to food after a cook doesn't wash their hands, and the pathogens are ingested when the food is eaten. Assuming that there's a mark available for each describe and each explain, and you can see we've given 6 potential marking points there.
So fermentation is one of your big passions, right?
Yeah, absolutely. That's a big part of what we do. Overall, there's a lot of spices and things going on here, but we're going to be doing a little root vegetable mix, sort of like a Oaxacan type of thing, but really my own sort of combination. We're going to throw in a little bit of salt, and unlike a proper lactic fermentation, we really don't need a huge amount of salt. You want to give that a little bit of mix right there. And then simultaneously as you do that, we got this lovely little golden bear honey that we're going to throw in too for sweetness. We're also going to throw in here a good amount of cumin. This is a mix I just made in the kitchen of anchos and chipotle, just regular ground cinnamon, nutmeg, and some fresh lemon.
I'm already smelling this kind of Mexican flavor profile.
All right, why don't you give that a taste and see where we're at. How we doing, Chef? We all right? It's starting to taste good?
It's incredible.
Fermentation is one of the more enigmatic processes in any chef's bag of tricks. It's not quite baking, nor is it cooking, but the unique alchemy achieved by coaxing the natural processes of decay and transformation can produce indescribably wonderful results. Mastering the basics may be easy, but achieving something memorable, let alone repeatable, isn't. It's a lot like opening a good restaurant. By some combination of vision, serendipity, and faith, something magical happens, like Duck's Eatery.
Good afternoon, Duck's Eatery.
Brother and sister Will and Julie Horowitz have been running this East Village eatery since 2012. In restaurant years, that's an eternity for a charming gem of a neighborhood place like this. But like thousands of restaurants across NYC and the country, Duck's had to announce that it was shutting down for good. COVID-19 has been brutal for the restaurant industry. As winter approaches, the prospect of mass closures is a near inevitability. Absent an overnight influx of government support, Band-Aid fixes like outdoor dining and partial capacity seating offer little hope.
You take care. Bye. And in a fun spin, our Diners, Drive-Ins, and Dives episode just aired. Son of a bitch.
So take me through the next 48 hours. I mean, this is it, right? This is the last hurrah?
This is it.
What are you guys doing tonight?
So tonight is our last brisket night. It's something that we started way back in the day. Nice and overbooked for it, which is super complicated to navigate during a pandemic. But really touching. I mean, people have come out of the woodwork. It's pretty amazing. We opened about 4 months before Hurricane Sandy hit. So we're sort of bookending our experience here with major events, which kind of feels right.
Looking back on these last few months, do you point the finger at anyone specifically?
I mean, this is just a huge loss. And you know, if I had it my way, the money that went to the airline bailouts, you know, would be distributed more evenly.
So tell me a little bit more about Ducks. Like, what differentiates you guys? Like, what did you set out to do with this restaurant?
So I think the biggest thing that differentiated us with the restaurant is that we didn't set out to do anything. I think we spent so long trying to piece together the funds and everything and screws and nails and wood to build this place that I think if I remember like the night before we opened, me and my other chef kind of freaked out because we forgot to make the menu. You know, when we first started smoking briskets in here and winning awards, we were sleeping on the tables just, you know, so we could be closer to that process and make sure we didn't fuck anything up. We were going to our farmers and butchers and saying, okay, what do you have extra of? What can't you use anymore? What's garbage right now? Give it to us. Let's figure out how to make it into something delicious by looking at these ideas back in time, like smoking and slow cooking and aging. That's sort of the beauty of these old techniques. And, you know, we did it like over and over again in the restaurant. And people thought we were being like, you know, really creative and, you know, artsy and shit. But like the truth is, we were survivalists.
I want to talk about the pandemic a little bit.
The places that you see here that are busy, they're still losing thousands and thousands of dollars a month. They're like, it's just a show, it's not for real. There's very few people that are coming out of this thing unhinged.
Are you angry?
Yeah, I mean, I'm pretty pissed off. I think from like the bottom to the top, our leadership has failed us. You know, there's been nothing for us. You know, and right now, we're all looking at the end of the barrel, down a cold winter most likely, and everyone's PPP money has, you know, dried up. And, you know, there's really no way coming out of that. What we're looking at is something far worse than that because we're looking at, you know, the 100-year-old places and mom and pop places shutting down. And that wasn't an issue in 2008. Let me get this. Hi, this is Duck's Eatery. Yeah, what country are you calling from? Oh, wow, that really means a lot to me. I really appreciate that a lot. My name is Will Horowitz. I'm the chef. I have to get back in the kitchen. We're smoking brisket for the last time tonight. But I really, really appreciate you calling from Italy, and I appreciate everything you said and how special it was to you. Have a good day and stay safe, okay? That was amazing. I was trying to see if I could speakerphone that. I swear someone was on the other line.
Wow.
And you can see these guys, we have like really kind of like a perfect bark going on to these.
Tonight marks the end of a chapter for the extended family that has come to call Ducks home. But for the time being, any bittersweet feelings would have to wait. The kitchen was starting to get busy, and folks were starting to arrive. Outside, it was beginning to feel like so many of the NYC restaurants I've come to love and cherish over the years.
Tonight was the last night they were doing their brisket special, so I knew today was like the end-all be-all.
Duck's Eatery is family for me. It is my life, it is my home, it's everything. All my memories are here are special. I got engaged here. I fell in love with my person here.
They really built a culture on this street. I've stopped in here, I think, every month since they've opened. And I love you guys, and I'm here for you, as always. On to the next thing.